Hi, my name is Lauren Louie, and I'm a research scientist at Lawrence Berkeley National Lab. And I'm really excited today to talk about my Nanopore metagenomics project of both European and North American coastal samples. So this is a picture of me in Matane Jane's lab at Northeastern University. I loaded 34 Promethean flow cells, as you can see in this picture on the right, and all of them turned out really well. I didn't get any air bubbles in them. They're all green. And so we did that all in one day, and that resulted in 5 terabases of Nanopore metagenomic sequencing. And so I'd like to talk to you today about how we got to this point. So my research is about the San Francisco Estuary, and this is a very, very important resource for the San Francisco Bay Area and actually all of California. So it comprises of the Sacramento-San Joaquin River Delta, Sisseton Bay, San Pablo Bay, and San Francisco Bay. And so this estuary supplies 2/3 of the drinking water to California. So Southern California draws a lot of its water from Northern California. And it's also very important for commerce, recreation, agriculture. And so it really behooves us to take care of this natural resource. But because of the millions of people that live in this area, you can also see how it can be it's highly impacted by human activity. So runoff from cities, agricultural runoff, wastewater treatment plants all go into the estuary. And so we really need to take care of this natural resource. So one of the things about the estuary that people have been surprised about is that it's not experiencing eutrophication. So an example of eutrophication is in the top right corner. And you can see how the algae is covering the water. And when that happens, that also causes hypoxia, which will kill fish and really cause the ecosystem to become destroyed. And so we really don't want that to happen. But the San Francisco Bay is actually experiencing high nutrient loadings that we understand that can cause eutrophication. But this isn't happening yet. One of the hypotheses about why this isn't happening is because of the turbidity from the runoff from all of the rivers into the estuary contain a lot of silt. And so that causes the sunlight to not penetrate the water and prevents the algae from growing, but we still don't fully understand what's happening. So although San Francisco has not been experiencing as much eutrophication as other areas, we have started to have more of these harmful algal blooms. So this is an example of a harmful algal bloom that happened in 2022. And thousands of fish died. And so the city of Oakland has been trying to mitigate what's happening by oxygenating the water. But the presence of more of these harmful algal blooms really indicates that the ecosystem is imbalanced, and we need to study what's going on so we can prevent them. So one of the things that we can do is study the microbiome, which is my research. And so I was talking about these nitrogen and phosphorus loadings. And so the microbes control parts of the nitrogen cycle that only they can control. And so we really have to figure out what they're doing, when they're doing. But we can't culture a lot of them in the lab. And so what we can do is sequence their genomes and look at their genes to try to figure out what they're doing. And so that's what I've been trying to do with environmental sequencing. So environmental sequencing is pretty difficult, and the way I like to explain it is through this puzzle analogy. So imagine that you have all of these microbial genomes, and they're like puzzles, and then you dump all of these puzzles into one box. But then you don't stop there. You dump all of the pieces in a blender, shake it up, and then try to reassemble all of the puzzles. So this is basically how environmental sequencing has been and why it's been so difficult. So an example of how difficult it is to assemble these genomes is if you look at the standards for high, medium, and low quality. So if medium quality metagenome assembled genome is only 50% complete, you can see how difficult it is to get a complete genome out. So these standards were developed from Illumina sequencing, but I've turned to Nanopore sequencing because the long reads help us get more complete genomes out. And so bacterial and archaeal genomes are circular. And so you can see on the right side, we're getting many more complete genomes out from using Nanopore sequencing. So this example I'm showing you is a groundwater metagenome, the exact same sample that was sequenced with both Illumina and Nanopore. And so you can see on the left side, the Illumina metagenome is much more fragmented and tangled. But the Nanopore one, we're actually pulling out these complete genomes. On the bottom, you can start to see some of the other circular elements that I highlighted that are smaller. These are phage and viruses and some plasmids. And so we can start to really look at the microbiome. So I've been collaborating with the U.S. Geological Survey to get samples from the San Francisco Bay. And they do monthly cruises to look at water quality. And I'm currently getting samples from 8 of their stations. And I'm going to talk to you about data from this pilot sample that we got from the South Bay. So this one sample we sequenced to depth of 150 gigabases. And to get the highest quality data we could, we developed new DNA extraction methods because you can't get long reads if you don't have long pieces of DNA, and also worked on the bioinformatics methods that were specialized for long read sequencing to really get complete genomes out. And this is on bioRxiv, but it's also in revision now in M systems. And so this DNA extraction method, you can see here, we're getting much longer pieces of DNA compared to other environmental samples that we've extracted before. This novel binning method, it's also actually a bin refinement method. We're able to separate strains. So in this example here, you can see that there's 2 very similar genomes based off of sequence identity, but they are different from each other. Because of the longer reads, that actually helps us with taxonomic classification. And so we decided to also see if we could classify every contig. So now instead of being limited by the length of contigs for classification, it's the databases that are really the limiting factor now. The other thing that we got from this analysis is that we assembled a complete pico-eukaryotic genome, which we didn't expect. And we also were getting chloroplasts and mitochondria. Because we were able to assemble large pieces of genomes and circular genomes, that's what the blue dots are in this picture, we were able to look more closely at strain level differences in abundance. So on this example on the right here, there's one star 86 that has some closely related other strains in the sample, but it is much more abundant than its neighbors that are closely related. Another thing that we got was complete genomes of these giant viruses. So a lot of these infect algae. So we do have algae in our sample. So maybe it shouldn't be so surprising that we have these, but it was really great that we got complete giant viruses from these samples. And so that's how we got to this point. So like I said, we were sequencing very deeply to get as many complete genomes as possible. And so that's how we got to the 5 terabases of data. We've been getting complete genomes from marine microbes that people have been trying to get complete genomes from for a very long time, like SAR 86 and SAR 11. And so if you thought 5 terabases was a lot, now we have 5 terabases of Baltic Sea sequencing. So we're collaborating with Stefan Bertelsen, and we have 20 sites that we've gotten samples from and have sequenced, and we're still analyzing them now. But it'll be really exciting to compare to the San Francisco Estuary because these are both two highly impacted estuaries from human activities. And so I'd like to thank Lawrence Berkeley National Lab for funding for this project, and Torben Nielsen, who's been working with me on extracting and processing and assembling all of this data, and all of our collaborators that really supported this research. So thank you to Nanopore for giving me the opportunity to present my research.